Are you there? He's not muted, is he?
He
No.
is unmuted, but
He's frozen.
Well, I see that he is unmuted. Dr. Rosen, we're having a little difficulty. We do not have your audio. We heard you for a millisecond. I don't know if it's possible. You may want to use the telephone call-in line if you're able to do so. He is unmuted. He just left to go call in. So Dr. Rosen very graciously offered to do this, though he just got off a plane in another state just a few minutes ago. So it's probably very challenging for him. But he should be back with us just in a moment. All right. Oh, he's muted.
All right, can you hear me now?
And
All right, finally, I had to restart the whole thing.
Sorry
So,
about that.
good, now I have to give a shorter talk. So my topic is blue-green algae, and I'm going to focus in on some of the potential human health effects and also the kind of research that I'm doing at Florida Gulf Coast University. Next slide. So first of all, keep in mind that cyanobacteria are in a family of algae. And what unites all the algae is that they have chlorophyll A. So every colored box in this family tree of life are organisms that are considered algae only because they have chlorophyll A. Cyanobacteria are bacteria, they're very primitive. Then you've got the euglenoids, the cryptomonids, the dinoflagellates. You got the red algae, the diatoms, and even land plants and animals are on the same family tree. But again, the point is anything that's colored in here has a nexus to being called algae. Go ahead, next slide. So that unifying feature is that chlorophyll molecule on the right. And this is a colony of Microcystis that I've blown up. You can see the individual cells and you can see the bacteria that are associated with this colony. You can see how tightly packed the cells are. And that morphology might tell us what species this is. Not always. So this is not your typical Microcystis aeruginosa because it doesn't have these big holes in the middle. Go ahead, next slide. They have a lot of pigments. They don't have just chlorophyll A, they have the phycocyanins. So hence you hear the blue-green algae part. They also have phycoerythrin, which is a reddish color. So whenever you see a bloom, and even under the microscope, they could look brownish because of the reddish color from the phycoerythrin mixing with the phycocyanin. And they have other pigments too, all there to capture sunlight. Next slide. And when they die, when people often say there's this turquoise staining of the shoreline, that's when the pigment, the phycocyanin, leaks out of the dead cells. And it's not a molecule they want to lose. They want to keep that molecule in the cells. So it's only when a bloom is dying off or individual cells are dying off that you see this bluish color coming out of a cyanobacteria bloom. Next slide. So part one, in my lab, we do a lot of work on morphology. In other words, what do these organisms look like? And again, even though the cyanobacteria are bacteria, they're a little more sophisticated than other bacteria. They can have a resting spore called an aconite. They can have a heterocyte. These are really the filamentous forms. This does not happen with the Microcystis, which is a colonial coccoid form. And to better understand what they're doing, in my lab, we try to isolate these organisms that come to me, pull individual filaments out of the water, and identify what they are morphologically, but then also we're doing work with DNA to sequence these organisms, because we really don't know which organisms are making all the different toxins. Some we know, Microcystis ferruginosa can make microcystin, doesn't always, but it can. But then you have some of these other forms. These are in Lake Okeechobee as an example. What toxin are they making? What toxin should we be screening for in Lake Okeechobee? Next slide. So part of the reason cyanobacteria is so successful is that they can regulate their own buoyancy in the water column. Not all of them can, but let's just say most of them can. Microcystis can. This is a Dolichospermum on the left. They've got this structure called the gas vesicle. Again, as a bacteria, they don't have membrane-bound organelles, but they've got this proteinaceous structure that holds gas, and they will float up first thing in the morning, photosynthesize, and the turgor pressure that builds up from making the molecules of sugar from photosynthesis causes this gas vesicle to collapse, and then they sink back down in the water. So there's this up and down movement. And when they go down into the lower part of the water column, especially at night, there's no light, so they start utilizing that sugar. It causes them to fill up these gas vesicles and come back up again. And oh, by the way, when they're down there, they also can pick up a load of nitrogen or phosphorus, really phosphorus that gets liberated from sediments in the bottom of, let's say, a lake or any water body. Next. So that's important to understand if you have a drinking water source. This is Las Vegas. And if you have a way of moving and having a different drinking water source, this is probably exaggerated. They don't have this kind of water right now. But as that bloom comes up and is buoyant, and if you're taking surface water, that's bad. There's an impact to drinking water. Next slide. Oh, and the wind blows them. Wind blows them like crazy. Look at this scenario from NOAA. These are real satellite imagery and an inferred cyanobacteria density. This is 2018, June 12th, not much you can see. By the 20th, there's a pretty decent patch out there. The 24th, 4 days later, and the 29th. This shows A, rapid growth, and B, them rising to the top and being moved around in a water body. This is a very large bloom. Next slide. So why do we really care? There's an ecological effect from low dissolved oxygen that causes big problems. Fish kills are really from that, not from this next topic, which are the cyanotoxins. There are basically three major groups: the liver toxins, hepatotoxins, the neurotoxins, the skin toxins, and the one I'll put on here is called BMAA, and it's basically an amino acid that could potentially link to ALS and some of the other neurodegenerative diseases. Let's go forward one. So how toxic are they? If you look at how they measure toxicity, it's micrograms per kilogram of body weight to have a lethal dose where 50% of the mice die. We don't do this on people. It's hard to get volunteers. But anyway, that's algae joke. So saxitoxin is a very, very potent toxin. 9 micrograms per kilogram of body weight is potent. You might hear of saxitoxin in the marine environment, very common. And we think evolutionarily, the saxitoxin probably originated with the cyanobacteria and migrated to the dinoflagellates. So racine is a common, well-known toxin from castor beans. Okay, it's much more potent. But look at saxitoxin and anatoxin AS, it's now called guanotoxin, are as potent as copratoxin. Microcystin LR, the most common form of microcystin we see out there, is less potent than copratoxin, but more potent than curare and strychnine. You get my point. These are very potent compounds, but it really depends how they get into you. Exposure routes are the whole story. These numbers are all from ingestion. So it's not from inhalation. We know very little about inhalation. We know very little about the concentrations and how much it really affects, and there are no guidelines for that yet. So digestion, ingestion of these compounds, drinking water is really the most common source. Next. So in my lab, we wanted to find out more about aerosolizations. I did a little takeoff of what Mike Parsons was doing, and I wanted a very controlled these are little animations if you want to push one of them to start if you can. I don't know. Yep. So, oh, it flipped on you, but we wanted to aerate and capture the gases coming off of a bloom and then quantify how much is really getting into the air, both on, number of micrograms of toxin per liter getting into our lungs. And from here, next slide. Well, you'll have to see if we can get to the next. Yeah. So we went into the same chamber that Mike uses, these artificial lungs, I call them. It's really an air impactor where you collect layers down, and it's supposed to mimic the trachea up at the top layer all the way down to the alveoli at the bottom layer, and trying to find out, okay, so how much is breaking through? It's not a perfect setup, these air impactors. It's not a sharp division in size of the molecules, but we now have a counter that lets us count in size the particles. We know how much toxin is in the water. We know how much is getting into these different layers. And one of the unique things we found is it's not just the toxins breaking out into this chamber. Whole organisms are. Whole organisms become aerosolized. And we do a lot of this with cultures, and we also do it with natural blooms. Next. So as far as understanding how potent these are, there's drinking water guidelines that EPA promulgated. It's 0.3 micrograms per liter for microcystin, and another compound is cylindrospermopsin, 0.7 micrograms. But as children younger than school age, and the exposure has to be for 10 days. And if you're older than that, you can take a heavier dose. So I don't know how they regulate that at the tap. I don't think you can. You've got to go for the more conservative. But again, these are not regulations. These are guidelines. It was released after the work that happened in the Toledo incident, Toledo, Ohio. Recreational guidelines came along a little bit later. We have 8 micrograms of microcystin, 15 for cylindrospermopsin. But the EPA said for these, it's instantaneous exposure. And what that means, if you're out in a kayak and you might fall into the water, or really children on the edge of the water where they might accidentally drink the water, that's why these are instantaneous exposures. And again, here's the link if you want to look up those different guidelines. And we follow that. And a lot of times in Florida, we're proactive. If we see a bloom out there, we'll post it early on before we even know there's toxins there. Next slide. So we have a nice grant from FDEP, an innovation grant for looking at preventive technology. We're working in the Pahokee Marina, and we're using nanobubbles. We have 4 nanobubble generators, and the concept behind it is published by someone else, where these small microbubbles, nanobubbles really, when they burst, we think there's something about their tension that it disrupts and might hurt the cyanobacteria. We're still finding out what's going on in Mother Nature, and we're also moving to doing this in a very controlled dynamic situation in the laboratory. And then we'll look at all sorts of physiological responses. We'll find out exactly what happens to photosynthesis, what happens to the colonies. Do they break up? Does it affect the toxins? We're gonna have a much more detailed study of that from the laboratory to go with the field work. Next slide. All right, I'm gonna wrap up. So remember, when you go to a water body, the lifeguard, the bacteriologist, the microbiologist, and me, the phycologist, is not on duty. And a phycologist is someone that studies algae. And when my folks were alive, they thought I was a psychologist. And I said, yeah, I studied the psychology of algae. All right, one last slide, just for fun. Remember that cyanobacteria are truly art. This is a gliotrichia, and that spray of green you see on the outside are the bacteria that live on this colony. And in the green in the center is actually the heterocytes. And the heterocytes, by the way, are where, if you have one, you can fix atmospheric nitrogen, N2, right out of the air. And so you never run out of nitrogen as a fuel. Still need phosphorus, but phosphorus, you get all the nitrogen you want from the atmosphere. All right, I am ready for questions.
Thank you, Dr. Rosen.